welcome to the Jack Westin MCAT podcast with your host, Ahmed and
Eric Olson.
Welcome back, everyone, to another episode of the Jack Westin MCAT podcast with Ahmed and Eric. Today, we're going to be looking at DNA replication, or we'll say replication in general, as we'll see a little bit later why I made that distinction. We'll be talking about eukaryotes and prokaryotes, as well as viruses. Before we dive into that, Eric, how are things? What's new? How's year 2 of residency going?
Things are good. I mean, it feels like year 1, because starting over in a new field, but got a month under my belt, which feels pretty good. It's funny, like some days I come home and I say to my wife, like, I think that today I got significantly worse than I was yesterday. And other days I come home and I'm like, I was getting things, I was diagnosing things correctly. And it's just, there's like so much to know. And I feel like I'm just right at the very front end of it. So it's still a lot, but it's fun to finally be kind of doing the field, doing the things that you want to be doing. So
right.
It's been a good time. Yeah. How are things for you?
Fair enough. Things are good. Yeah. I rented my U-Haul. Like I got my booking in. So yeah. Moving days coming up soon in 11 days.
Oh yeah, that's really soon.
Yeah, I'm excited. I actually my apartment building is so cute. It's like a brand new building right in the middle of downtown, and like they have a bunch of these raffles. And so I'm like, I got nothing to lose. I'm just gonna throw my name in all of these raffles and see if I get anything. And like they're pretty cool perks. Like one of them was like a sweet experience at a Blue Jays game. One of them was like a batting practice experience. Like they're different other giveaways.
These are raffles that your apartment complex is putting on?
Like the condo building, yeah. For residents of the building. I'm like, the building opened a month ago. There's not that many residents yet. So like,
you got a good shot.
I've got great odds here. And I ended up winning like the suite experience at the Blue Jays game. And
that's awesome.
When I entered, I was like, if I win anything, I'm selling it and putting it towards like rent or tuition.
Yeah.
Then I won that one. I was like, you know what? Like I might want to go to this one. And so
what game is it?
It's a baseball game. It's the Blue Jays hosting the A's on next Friday. So yeah.
When I was a kid, I played some baseball, like a little kid, like probably from like age 6 to 8 or 9 maybe.
Yeah.
And the team that I was on was they had like the teams in the league were all based on like major league teams, and mine was the A's. So I've always been an athletics fan.
Yeah, fair enough. It's funny when I was in Little League, the team, like the one year I was the Red Sox, but the second year I was the Blue Jays, and I was the Blue Jays the year before I moved to Canada. Yeah. So it was just like very poetic in that respect. But yeah, exactly. But I'm stoked, man. It's like I've always been, like whenever I go to sports games, unless it's like a real like something that like I am like very, very passionate about, I'll just kind of get whatever seats make sense.
Yeah.
But yeah, no, this is gonna be cool. It's I think it'll be neat too to meet like other residents in the building. Because they like there's like multiple winners from like multiple of the condo buildings that like this like property management company owns.
Yeah.
So yeah, it'll be cool. I'm excited.
That's awesome. So beware though, because when I started residency and I did, my intern year in Atlanta, and they put on this thing for us when we first got there, that was like, we're going to take everyone to a Braves game.
Yeah.
Baseball games, like, I feel like you go to a baseball game and like you're kind of watching, but you're kind of not, like the action's not nonstop. There's a lot of kind of taking around that happens, but they like got us all tickets to a suite as well, or like a box or whatever. And it was cool. It was a cool experience, but like no one else wanted to watch the baseball game. And I was like, I didn't want to be like the odd man out. These were like the people I was going to work with for the next year, and I was just starting, and I didn't want to like weird impression, but I was like, this is fun and all. Like I love hanging out, but like, can we watch the game? You know?
Yeah, yeah.
So you never know what you're gonna get
I
with
think
that.
baseball is very networking friendly.
Yes,
Unlike
yes.
basketball or soccer. Like soccer is probably the least networking friendly, just because there's
Like
like
you're watching.
yeah,
Yeah,
like time's
yeah.
always ticking. The nice thing is though, and like this is gonna sound bad, but like these are people that are living in my building and other like owned buildings by the same company. Like these aren't necessarily people that I have to deal with day to day if I don't really want to. So
But you
a little,
can if you like them.
yeah, the option's there, but it's not a requirement by by any means. But no, it's cool. I get a +1, so I'm taking a buddy of mine who lives in the city. And so it's gonna be a lot of fun. But yeah, we'll see. Maybe I'll catch a foul ball.
Yeah, maybe so.
We'll
You'll have
see
to
what
let us
happens.
know.
But in any case, today's topic, as we mentioned earlier, we're looking at replication. This is a topic that there's a little bit of confusion depending on, the class of organisms at hand. For anyone that's taken like a cell bio or molecular bio, like the level to which you'll go into these things on the MCAT is significantly lower or shallower, we'll say. Like I remember when I took molecular genetics or something like that, it was like very, very specific, like different types of polymerases and, different you know, cofactors and different, whatever. Not a thing on the MCAT. Like, you know, the polymerase is like 1, 2, and 3. Like I would know 2 maybe, but even then, like rarely do I see those specific different types of the enzymes mentioned. But
Yeah,
just something to keep in mind as we go through. Um, but the general function, obviously, of replication is to create more identical genetic material to enable, the organism cells to replicate, divide, and then, survive essentially. Um, and so, Eric, curious, when you kind of think of replication in the context of the MCAT, like how did you study it? Like what were your points of emphasis? That kind of thing.
yeah. I mean, kind of a loaded question for me, just because I think I've mentioned this before, but like, tell me, am I lagging at all?
It's not too bad now.
Not too bad. Okay, if it starts to become difficult to understand me, let me know, and we'll try to troubleshoot a little bit here. It's kind of a loaded question for me because I took a molecular biology class as a freshman, and it like made me change my major to molecular biology. So this is something that I really enjoyed as an undergrad, and I think it's important for your understanding of disease at the end of the day too. So it's not just so like you said, I'm at some of the specifics of like these different binding proteins and things like that may not play into your practice of medicine so much, but an understanding of DNA and the way that it works and its importance in the human body, I think certainly comes into play, especially as we talk about cancer. Um, when I was studying for the MCAT, I think the important things to note, honestly, there's some of these enzymes and proteins that participate in the replication process that are more important than others. Um, I don't know that we need to emphasize them all, but there are a few that I would make sure that you kind of know and are familiar with kind of their function, because sometimes you'll get a question where they'll be, talking about some, like they could be telling you about a lab technique, for example, that you like start with a double-stranded DNA and you do something to it, and then you end up with all these single strands, and it would be like, what enzyme is performing this function? And so to a certain degree, you do need to know some of the enzyme and kind of what they do. But I think the big part and the part that they're going to try and trip you up on is like the actual like mechanics of replication. Like what's happening when you have replication going on to a strand of DNA. And so understanding, the way that it kind of unzips, and then that you have like a leading strand and a lagging strand. Some of the specifics about the DNA or the nucleic acid molecules, I should say, and like how DNA has to be formed, 5 to 3, and how it's read 3 to 5, and things like that. And so we'll jump into some of those specifics. But I think like that active process of replication to me is probably where the most of your money is going to be made as far as the MCAT goes. But I don't know, what are your thoughts, Ahmed?
Yeah, I think you hit the nail on the head. There's certain key enzymes that we need to be aware of. And just the general structure of DNA, how that differs from eukaryotes to prokaryotes is another thing that we'll talk about. There's, additional enzymes that are required in, depending on the structure that the DNA assumes. And so we'll get into all of that. I think to start, right, we know DNA is double stranded, right? And so to be able to access those nucleotides, right, those nitrogenous bases specifically, which are the rungs of the ladder, so to speak, we've got to get into the strands. And so we kind of have to separate those strands, right? And so that's where our first enzyme comes in, and that's the helicase enzyme.
Yeah.
Helicase, I always just kind of remember it as like A's tells me it's an enzyme, and then the prefix tells me what it's breaking down. So it's kind of opening the helix. And so to your point about leading and lagging strands, right? If you think of, your DNA strand as, this ladder, you're putting your helicases kind of back to back and letting them run in opposite directions,
Mm-hmm.
right? And so this opening that you get is what we'll call the replication bubble. And so as that helicase is moving, its goal is to just separate the DNA strands so that other enzymes can come in and do their job, right? But
Yeah.
the DNA is not going to stay separate on its own. That's why we have these things called SSBs, single-stranded binding proteins. And the name kind of tells you everything you know. It binds to single strands of DNA to keep those nitrogenous bases separate. Okay. If you think of the helicase moving down the strand, right? Imagine you have a rope and you have like the two, like, what do you want to call them? Like the strands of rope or little fibers of rope that are coiled, and you pull them apart. There's going to come a point where you've created so much tension that you can't pull anymore, right? And the DNA is the same way. And there's a specific class of enzymes that are responsible for preventing what we call, supercoiling. Those are called topoisomerases. Basically, what they'll do is they'll snip the supercoil, release tension, and then like reattach basically that snipped part. In my experience, and feel free to chime in here, Eric, I haven't seen topoisomerases much on the MCAT.
No, I don't remember ever seeing them. I will say it's like not a bad thing to know, just because it will come up in medical school, like and I mentioned like a lot of these things have to do with our understanding of oncology. And honestly, a lot of our understanding of DNA and replication, the way things work, comes from like cancer research. Like we started learning about cancer, and that's how we developed an understanding of like this normal physiology. But there are certain chemotherapies that a lot of chemotherapies will target like different cellular pathways. And one of the things that we target, because, and I know this isn't an episode about cancer, but just because it's interesting, one of the ways that we try to target cancer cells above other cells is that they replicate at a faster rate than most other cells. And so one of the medications or some of the medications that we'll use will target the topoisomerases because they're necessary for DNA replication. And so if you have cells that are, dividing quickly, then they do a lot of DNA replication. And so if you target those enzymes that are involved in replication, then it's like a little bit more specific for cancer cells. But in a similar vein, and again, maybe this doesn't matter, but just for interest's sake, some of the other cells that kind of divide more rapidly in your body are like your hair cells, the cells in the hair follicle, and then the cells in the GI tract. And so that's why, when a lot of times with some of these chemotherapeutics, we'll get, hair loss or alopecia, and we'll have like GI side effects like nausea, vomiting, stuff like that.
Right.
Because, those drugs will target these rapidly dividing cells and these, DNA replication enzymes. And so all the cells that are dividing are going to be affected negatively. So it's kind of an interesting tie-in. And again, like when you take some of your first board exam, Ahmed, you'll be studying these things of like, oh, these are the topoisomerase inhibitors, and like here's what we
Right.
might use them for, and things like that.
Looking forward to it. Thankfully, it's passed down now, so very, very
Yeah,
grateful for
sure.
that. But no, I think it's always great to have those connections because number one, it makes learning more fun, and number two, it gives us a sense of purpose. For like, okay, like why does this actually matter, right?
Yeah,
And granted, there's some things on the MCAT that once you're done the MCAT, they just don't particularly matter anymore, right? Like alpha and beta decay, unless you want to work in, radiation, even then, like probably not super high on, your list or, you know, kinematics and projectiles. And like, but I think obviously like DNA replication and its connections to oncology, as you mentioned, it's a huge, huge part of, modern medicine. I think, finding effective ways to treat, prevent, cure cancer are one of the biggest, or is one of the biggest objectives, we'll say, of, medicine now. But like I said, always good to have those connections and applications. And continuing along the story there with replication, we have the topoisomerases basically preventing supercoiling, allowing those helicase enzymes to keep moving. And so now we're really opening it up for DNA polymerase to come in and essentially start the replication process.
yeah,
One of the caveats with DNA polymerase specifically, as opposed to RNA polymerase, which we'll use in transcription, is that DNA polymerase can't just drop nucleotides.
yeah,
It needs a free 3' hydroxyl group in order to do this. And so that might be a good time to take a second and just review the structure of a nucleotide, right? A
yeah.
nucleotide
Beautiful.
has three components, right? We've got our nitrogenous base, we've got our sugar, we've got our phosphate group. I
Hmm.
think the easiest way to think about it is to think of the sugar as the central part of the base, right? Or the nucleotide, excuse me. And it's a 5-carbon sugar. That's why we say the pentose phosphate pathway is critical in nucleotide synthesis because it's creating the pentose, the 5-carbon sugar. Carbon 1, we've got the nitrogenous base. Carbon 3, we've got a hydroxyl group to which the phosphate group on carbon 5 attaches. Carbon 4 is kind of irrelevant, all things considered.
Yeah.
Carbon 2 has applications with respect to DNA and RNA. DNA deoxyribonucleic acid does not have an OH group on carbon 2, whereas
Hmm.
ribonucleic acid does. So just some things to keep in mind there, but we need a three prime hydroxyl group for these DNA nucleotides to be added. And so for that reason, we actually need an RNA primer to be laid down, and then DNA can be added to that primer. And so that kind of gets us into, the whole leading and lagging strand situation where we've got helicases moving in two different directions, but we know, as you mentioned, Eric, that strands can only be read in one direction, right? They have to be read 3' to 5' so that they can be built 5' to 3'. And so, do you have a good way of maybe visualizing, those leading versus lagging strands and like what that means, like why that arises?
Yeah. I mean, I think I will say, like, and you touched on the structure of a nucleotide. I think a lot of us are kind of visual learners, and so I think it would not be unhelpful to go and like look at
Yes.
a depiction of a single nucleotide and like see what it looks like and what we're talking about. You have the phosphate that sits kind of on the outside, and has a negative charge, right, because of its oxygens, and so that's like where you get the negative charge of DNA from. Like I would know that's like a little factoid I would know as well.
Yep.
Um, and then, like you said, I don't know if you really need to know, like, if you really have to have a perfect understanding of like where these different carbons are on the pentose sugar. But I think you do need to know exactly like you said, Ahmed, like the three carbon for specifically, I think you need to know is like this is where the hydroxyl group is. This is where we're adding
Yeah.
on the next link in the chain, right? Um, at the end of the day, like it takes so many like it's funny because you think of polymerase as like, oh, polymerase is like doing replication. Polymerase is the enzyme we need.
Yeah.
But like it needs so much help. You know what I mean? Like you
Yeah.
could imagine a world in which like polymerase could just come in and like scan the DNA and just like develop its own thing. Like no need for splitting it
Right.
into two different strands, no need for the single strand binding proteins, no need for topoisomerase. But that's just not how it works. Like polymerase is at the end of the day, like it does the replication, but like it needs this whole concert of other proteins. And so I think it's important to like remember that and kind of that's how I would frame it in my mind is like polymerase is kind of the star of the show, but
Right.
it needs so much help. And the way that I would think about the leading versus lagging, I think like you just have to learn the leading strand first. You have to learn about the strand that is going to be made from the free 3' hydroxyl moving forward. One of these strands is just going to go until you get to the end of the DNA. It's just made
Right.
in one big long fragment, and that's the easy one to understand, right?
Yeah.
Just like you said, you're putting down with RNA primase, or I guess not RNA primase, but primase. You're putting down like a small chunk of nucleotides that allow you kind of the scaffolding for your polymerase to run.
I think
And so,
scaffolding is a great word for that because that's
yeah.
exactly what it is.
And so then your polymerase will kind of take that and run with it. And when it has that free 3' hydroxylase, then it will just kind of go indefinitely. The
Right.
caveat is that, your DNA always has to be built from 5' to 3'. And so you're always
Correct.
gonna put down kind of your 5' onto a free 3 hydroxyl group and continue to add those nucleotides onto that strand. Where we run into trouble is that if you can imagine, and again, this in particular, I think is a really good. The nucleotide, I'm plus minus on if you need to look it up and really see what it looks like. But this
Right.
particular process, I would look up and either see a video or an image at least, because it's a little bit harder to understand conceptually without looking at it. But
Yeah.
because you can create a strand from 5' to 3' on one side, we'll say it's maybe the bottom side that we're looking at. Then on your top strand, the strand is kind of running in the opposite direction. And so you don't have a free 3 hydroxyl group on that side that you can move forward because you would be trying to create a strand from 3 to 5, if that makes
Right.
sense.
Right.
Polymerase doesn't run in that direction.
Yeah.
So kind of what ends up happening is that as you open up that, what do you call it, replication bubble? I
Yeah.
always, like replication fork is kind of the way that, and I think you could still say replication fork for like the corners, and then you kind of have a
Yeah. Yeah.
bubble in the middle where all the machinery is. But
Exactly.
what happens is because as you open up that bubble, then you have more space to instead of being replicating in kind of that 5 to 3 forward direction, you're going to have to come backwards along that replication bubble to continue to create things in a 5 to 3' direction because that's the only way that DNA polymerase will work. And so you end
Right.
up having, you only need one primase for your leading strand. You only need
Yeah.
one RNA primer because, again, you're just going to go indefinitely and continue to add nucleotides to that strand. But what happens with the lagging strand, and you can imagine, if your replication fork is like this, then your RNA or your primase would come in kind of at the corner here and lay down a couple of nucleotides, and then your DNA polymerase would come in and start to build backwards along that opening replication bubble, and build in some more nucleotides. But it's not going to last forever because you're going to run into the other side of the replication fork over there. And so in the meantime, your replication fork continues to kind of open and unzip the DNA strand down the line. And so your primase will come back in and put down another primer down the line. Your
Right.
polymerase will fill in the gap, and that will continue to occur. And so the leading strand, I think, is the easy one to understand, and that's kind of how I would frame things initially.
Yeah.
And then you kind of fill in the gaps with those with the lagging strand, and the each of those segments of DNA are called Okazaki fragments. Like that's something that could be helpful to
Right.
know.
I
But
think
what other tips do you have on it?
I was gonna say there's a good analogy that kind of came to mind for me. Um, and so I would think about, leading versus lagging strand, like wakeboarding versus snowboarding. What do I mean by that? If you're wakeboarding, you're just kind of getting pulled by the boat,
Yeah,
right? So once you're up, you're going as long as the boat is going,
you could go forever. Yeah,
right? And so that's kind of your leading strand where the boat is your helices and you are the prokaryotes. Okay. Snowboarding, though, you can only snowboard as far as the chairlift took you up, right? So the chairlift takes you up, you snowboard down. You go back on the chairlift, you can snowboard again, right? So the chairlift in that analogy is your helices. You're kind of letting it take you some distance that you can go down. And then
yeah,
once you're done that, you get on another chairlift, it moves you forward, you come back down, kind of thing. And so every like run, we'll say, would be an Okazaki fragment because it's just
yes,
a disconnected
a
segment of
segment. Yeah,
snowboarding, right?
exactly. I
Whereas
love
when
it.
you're wakeboarding, you're going, right? There's no segments. And so that can be a helpful analogy to try to compare and contrast the two. As far as the depth of understanding with them, I would say familiarity trumps mastery in this case. Like I would
Yeah.
know the terminology, I would know what an Okazaki fragment is. I wouldn't, prepare to write a dissertation on it though. I think where you're going to make your money with replication is knowing, the big enzymes. I would say helicase and polymerase are the two big ones. I would know that polymerase and ligase have proofreading ability, right?
Hmm.
So you'll want to know that with respect to different proofreading mechanisms like base excision repair or mismatch repair, etc. Uh, I would say I lost my train of thought. Knowing that you need the primers, I think, is definitely important. Knowing those three major carbons in the nucleotide, I think, is helpful. But yeah, what I was going to say was where you're going to make your money is also knowing the differences between, eukaryotic replication versus bacterial replication.
Well, Ahmed, before we jump into that, if we can, I would
Yeah.
also say, like, let's just talk about the binding of the nucleotide bases to each other. Like, what are the different
Yeah.
base types? I was gonna say earlier, um, I remember getting a question, and I can't remember where it came from, but I remember getting a question that had something to do with like some animal or organism or cell or whatever like was generating something with phosphorus in it, like it
Okay.
was creating some molecule and it had phosphorus in it. And the question was like, where did this, it must have been some sort of like carnivorous animal, because I remember the question, and I'm not doing it justice, but it asked something like, where did the phosphorus for this probably come from? And the correct answer was like, DNA from like some animal that it ate. And I
Right.
remember seeing this question and being like, I guess I didn't really like know that we ate DNA. You know what I mean? Like it doesn't
Yeah.
feel like something that we eat. You know what I mean? Like you think like, oh, well, we cook our food. Like maybe the DNA like escapes somehow or something like that. I don't know. Maybe that's like a funny thought, but I remember that's what I thought.
Yeah.
But really, like when you think about biomolecules, like we think about fats and carbohydrates and proteins, and like
Yeah.
those are the three major biomolecules. But a lot of times we'll kind of also have a fourth category, which is nucleic acids.
Yeah.
And so they are like a major part and kind of a distinct entity within these different biomolecules that we talk about. And like we said, they have these different components like the phosphate and the sugar, which could theoretically maybe be considered a carbohydrate. And then they have this nitrogenous base, which, you have a lot of nitrogen in proteins.
Yeah.
But they are kind of this like weird, distinct entity that it's worthwhile to be kind of a little bit more familiar with. And I think if you're thinking about like your macros and what you're eating, like we don't really consider DNA in any or like nucleic acids in any of those things
No.
because they're broken down in a very specific fashion. And like compared to like the amount of macromolecules in the rest of the things that we eat, like the amount of DNA and RNA is like very minimal, right?
Right.
But that said, like just in case you get this question, we do eat nucleic acids.
Yeah.
And I think it's worthwhile to kind of understand the different binding of kind of purines and pyrimidines to each other, and which ones
Yeah.
are stronger bonds, or like which ones have more bonds and things like that. Can you
Yeah.
touch on some of?
Yeah, so we know that a purine is, I always remember short word, bigger structure. So purine has two rings, pyrimidine has one ring. I've seen questions where, like on occasion, where you'll be asked to know the structures of the nucleotide. It's possible, it's not highly likely. And usually knowing whether it's a purine or pyrimidine can narrow it down to two for you. And
Yeah,
at that point, I
absolutely.
think that's more worthwhile than trying to memorize loop structures, because I think that time could probably be better spent elsewhere. But in any case, adenine and thymine will base pair together. Cytosine and guanine will base pair together, or adenine and uracil, depending on if it's DNA or RNA.
Yeah.
I remember pure as gold and cut the pie. So purines are adenine and guanine, and then cut the pie. So pyrimidines are CUT, cytosine, uracil, thymine. Makes sense that uracil and thymine are the same because they both bind,
They
or they
both bind to
both
adenine.
complement adenine. Um,
Well,
and
this is a
then
bond, right? Like it's a hydrogen bond, I think, right?
yeah,
So
it's the attraction between the bases there. Um, and so what I would say is know that there's three hydrogen bonds between between guanine and cytosine. There's two between adenine and thymine, or adenine and uracil. There's a question that I saw, I won't say where, so I don't spoil it for anybody, but it's asking like why, a GC ratio that's higher means it's more stable, and hydrogen bonding wasn't one of the answers. You may also see this concept of pi stacking interactions, which is just like, aromatic behavior. And so there's a higher overlap, we'll say, different electron clouds. Uh,
yeah.
beyond
And
this,
when you get like if you're doing like a PCR, like I worked in this lab and we would do PCR sometimes, and when you're like calculating the different temperatures, we should probably talk about PCR at some point, but there are different phases to PCR. Like there's an annealing
yep,
phase where you get kind of the different segments of nucleotides to fit together, and then there's a replication phase where you have like an extension of the thing, which is basically right what we're talking about right now, and then
right,
you have like a melting phase. Anyway, the temperatures at which you're going to be doing these things is dependent on like the CG content of your strand. Because if you
right,
have a string that's like exclusively, C to G bonds or C to G, complement, then each one of those is going to have three hydrogen bonds, and it's going to be harder to break apart. You're going to
right,
need more energy, a higher temperature. Whereas if you're looking at just A to T, then you can break it apart a lot easier because they only have two bonds between the two of them. So you have to like take that
right.
into your calculation. So it does play a part. I don't know if I've seen a question on that, but I think that sort of understanding would theoretically be fair game.
Yeah, and just knowing which primers would be more, temperature or thermostable, right?
Yeah,
Stable at higher
absolutely.
temperatures, I think, is just an extension of knowing the GC content. Um, and yeah, there's a lot of like little sort of tidbits of testable info there, knowing that, PCR just creates, lots and lots and lots of copies of, the same, DNA strand, right? Knowing the subtleties of GC content and how that translates to stability. Uh, and I think that's an important connection to be able to make as well when it comes to DNA and its structure and replication. Um, Anything else there
Yeah,
that you want to get
there's
to?
one other thing before we get to prokaryotes and eukaryotes that I think is helpful to touch on, and that's telomeres.
Yeah.
And I remember when I was an undergrad as well, and I was like looking for a lab to join,
Okay.
and I applied to all these different labs, and like none of them had room for me. I
Okay.
remember because there were some that I was really excited about, and some that I was less excited about. Um, but one was a guy who had like I can't remember his story exactly. I think he had been a researcher like at Hopkins or something, somewhere like that was a big deal.
Okay.
And he had like done he had done like telomere research, and it was I don't know, it was about aging or something like that. And so we'll talk briefly about like what aging has to do with telomeres, but I remember like I was very excited about the possibility of working on this stuff, and like I thought it had implications for aging and dementia and stuff like that. And he like didn't have room in his lab, so it didn't work out. But but basically, like the idea with telomeres is that they get short. The short story is that they get shorter like the more times you replicate.
Okay.
And really, if we think back to kind of this discussion we were having about the mechanics of replication, like the way that we were presenting it is like, with a replication bubble, and like we're in the middle of a strand of DNA. But at a certain point, like you have to consider, like you're going to get down to the end of this strand. And like what happens at the end of the strand, on our leading strand where we just put our primer and then we're kind of just going indefinitely, and then you just get to the end of the strand and you replicate the whole strand, and that's kind of the end of it. And there's, you don't really have any issues. Um, but if you're thinking about going in the opposite direction, the way that we've been doing things is we have to place a primer, and then we can replicate from 5 prime to 3 prime. When we get to the very end of the strand, the only way to replicate the end of the strand is by placing a primer at the very end, kind of like the very tip of that strand. And, I don't know, Ahmed, maybe like how many nucleotides long a primer is, but, do you know? I see.
Yeah. I'm not 100% sure. I know with respect to the telomeres, it's like the same sequence of like 6 nucleotides repeated just over and over and over again.
Yes.
Both of those two things are probably beyond the scope of the MCAT, though.
Okay. Yeah, no. All I'm saying is like, as far as a primer goes, like imagine it's, 10 base pairs long, 10 bases long. You're gonna
Yeah.
have one primer that sits at the very end of the
Yes.
DNA strand. And you can start at that 3 prime hydroxyl group and start to replicate and go forever, but you can't really get beyond that
You
because your
can't get there.
primer, your primer is taking that up, and you cannot replicate in the 3 prime to 5 prime direction. And so
Yeah.
every time you replicate at that end point, you're going to have this little bit of strand that gets cut off. 10 base pairs, was just like a total, just for the sake of example.
Right.
But you're going to be losing 10 base pairs or whatever the length of the primer is every time you replicate. And so, as Ahmed said, these telomeres are constructed of this like six base sequence, and I don't know exactly what it is, but it's not a coding region. It's not really important for the functionality of the DNA, but it's kind of this protective measure that allows us to keep the important parts of the DNA, which are kind of in the middle of the molecule, while we like kind of chip away at those end pieces, which are those telomeres. And so there's some thought out there, and I don't really know what the current thought is either, but I remember when like 5 or 6 years ago when I was an undergrad, that this was like something people were talking about is like the shortening of our telomeres with age every time that our cells replicate. Is that contributing to aging in some fashion? And another tie-in to cancer, one of the ways that cancer kind of induces like the immortal uh, characteristic of its ability to replicate without like ever stopping is that it can upregulate this enzyme called telomerase, which is like an enzyme that will place extra telomeres and extra base pairs on the ends of a DNA molecule so that you don't get into the important parts. Um, I don't know how much of this, maybe you could speak more, Ahmed, to like what of this is most important to understand for test day. But I think telomeres are certainly fair game. Like, what is their function of kind of just protecting things? How does telomerase work? And
Yeah.
things like that.
And so telomerase, like you said there, Eric, it's basically extending the end of the telomere so that the chromosome doesn't shorten, right? And to emphasize, this is only relevant with linear chromosomes because if it's circular, as in the case
Yes.
of DNA, right, it loops around, you do have that 3' hydroxyl. So telomerase is unique to eukaryotes because of their linear DNA structure. But you actually see it in some cancers where telomerase will be overactive, right? And what
Hmm.
happens is that prevents, natural cell death, or what we call senescence. After a certain number of replications, right? Those telomeres shorten and shorten and shorten. Now you start getting into coding parts of the DNA and the cell dies, which is a good
Yeah.
thing, right? Because you want it to die after a certain amount of, or like you want it to stop replicating after a certain amount of time. We call that the Hayflick limit because over time mutations accumulate, and the longer you let a cell run, the more likely it is to inevitably become cancerous, right? After a certain number of mutations accumulate. And so overexpression of telomerase is a bad thing because it allows those mutations to continue to accumulate unchecked. The one exception to that is our germ cells. Our germ cells do express telomerase. Otherwise, the entire species would come to a halt within a generation or two. Uh, so we need those germ cells to have that telomerase activity so that we can have, a full linear chromosome to pass down to our offspring. But as far as the MCAT goes, I would know that telomerase is eukaryotic only because of the linear structure, but I probably wouldn't know much more than that. Uh, and that's probably a good segue into, some of the other major differences between eukaryotes and prokaryotes. I think we touched on the structure there is the big one. The fact
The
that,
structure being, yeah, being that in eukaryotes we have linear DNA with kind of a
yeah,
start and point, and in prokaryotes it's a circle, or a lot of times we'll refer to them as plasmids.
exactly. Yeah, it's circular, double-stranded still in bacteria. Obviously, no nucleus in bacteria, no membrane-bound organelles, etc. And that might be a conversation for another day. But really, bacteria to eukaryotes, like that's the major difference. They both replicate. They both have the same nitrogenous bases. It's just whether it's linear or circular where you get a little bit more variabilities in the viruses. Uh, and so with viruses, you could have single stranded, you could have double stranded, you could have DNA viruses, you could have RNA viruses. And that kind of lends itself to one of three major groups of viruses. And that might be where we wrap up is, negative sense, positive sense, and retroviruses. And Eric, I know for me anyway, when I was studying this the first time, I was like, positive sense, negative sense, none of it makes any sense. It was it was something that I really had to work at to figure it out. Really, I'm curious. Like, do you have any inside info onto maybe how to simplify those terms?
I honestly, I don't. I feel similarly to you. I worked really hard to try to understand it, and I don't ever really feel like I got a great grasp on it, honestly. So I would bounce it back to you. I don't know if I have great advice on this particular
Yeah.
topic.
Well, so I always thought about like positive sense makes sense. So that's an RNA virus that can just come in and translate, you know, its transcript and create viral particles and just, replicate through the host, right? Because viral replication is not what we know from prokaryotes and eukaryotes, and that it's dependent on a host in the host cell machinery. So positive sense, the mRNA comes in, gets translated, and away we go. So that makes sense. Negative sense to me doesn't make sense. And so that one, the mRNA comes in, you have to transcribe a complement to that mRNA, and then that complement gets translated. And so for me, I was always like, well, negative sense, like, why not just like have the right mRNA entering the host? So that was kind of how I tried to frame that. And then reverse transcriptase was the one that made the most sense to me because it's all in the name, reverse transcription, right? So transcription is DNA to RNA. Reverse transcription is RNA to DNA. So you have this RNA Now you're reverse transcribing into DNA. That DNA then integrates into the host genome. And that's why retroviruses are so difficult to treat, like HIV, for example, because it's part of your genome now. And so it can be very, very difficult to cure it, per se. It's more so managing the symptoms. But that was kind of how I remember those three major types of viruses based on their mechanisms. Not sure if that makes perfect sense, but that's what worked for me.
Yeah, that's helpful. I think as far as retroviruses go, I'll just say, I agree with you. Like we certainly can't cure like HIV, I think is the prototype of like a retrovirus. And one thing that may be helpful and maybe will not be helpful, but one thing that we know about HIV is like it's one of the things we use when we're trying to insert like a sequence of DNA into a host genome. So like I worked in a lab. I may have talked about this at some point, but I worked in this lab. We were working on like tumor targeting T cells, if that makes sense. So we were like
Okay.
trying to like
Yeah.
develop certain T cell receptor regions, and so we were like trying to code out like the right amino acid sequence to get this T cell receptor the way we wanted it. And the way that you like get a code, like a sequence of DNA that you want into a eukaryotic cell is you like will encode basically an inactivated HIV virus to like go into a genome
Right.
and insert this specific sequence that you want. And so it is something that we use. It's like the prototypical retrovirus, and we certainly can't cure it. But it's also not just managing symptoms. Like we use antiviral medications that target like the mechanisms that a retrovirus uses to insert that DNA. So like there are proteases that we use that target some of the proteins that are involved, and like there are certain like nucleotide analogs that we use that help to like inactivate the machinery that an HIV virus uses. And so like we do have some understanding, and like we obviously like we can treat people so that they get to a point where they don't have symptoms, and like their white blood cell, really what HIV does, we don't have to get too much into it, but like it really tanks your T cell count. And that's why you get infections, is like you don't have any T cells basically. And
Right.
so like with the treatments that we have now, we can make it so that people have T cell counts that are akin to what you would see in a completely healthy individual. So we do have treatments
Right.
that are really effective. But we don't have a cure. Like you said, it's not like you take this antibiotic for 3 weeks and then you're good to go. It's like you take these antiviral medications for the rest of your life.
Yeah.
And so it is kind
Yeah.
of once it's in your genome, once it's in those cells, then you can stave it off, but it's part of your DNA. As we know, like DNA is really, really stable. It'll replicate with very high fidelity. That's not really something we talked about, but
Right.
the rate of error in DNA replication is like what is it, like it's like 1 in 100,000 bases or something like that. I don't know.
Yeah.
And then we have
Yeah.
proofreading ability on top of that, like you mentioned earlier. And so like we don't have a lot of errors when it comes to DNA replication. It works really well, and that's how you see like people that uh, have stable DNA throughout their lives. That's one of the more stable kind of biomolecules that we have and that we talk about.
Yeah. And I think the last thing I'll add just on that point there, Eric, in a connection to your future as a dermatologist, one of the most well documented mutations, little guest appearance here. Um, One of the most well known mutations on the MCAT are the inductions of these thymine dimers, right, from UV radiation. As an MCAT student, I would be aware of that. UV light exposure, right, sunlight exposure causes these adjacent thymines to bind to each other as opposed to the complementary strand. And so we need base excision repair to resolve that. And so, yeah, I would just know that as a med student as well, or sorry, as a MCAT student as well.
Yeah,
Um,
I actually just, sorry to interrupt, but I just had
yeah.
a lecture last week on like on like carcinogenesis or cancer development in the skin. And one of the questions that they asked during the lecture was like, okay, yeah, what kind of mutations do we often see in like UV damaged skin that leads to, this sort of cancer? And I
Right.
was like looking around, like waiting for someone to answer, and no one knew. I was like timing timers. And they were like, yeah,
Yeah.
yeah, yeah. And I thought, well, it's a good thing I remember some things from the MCAT. This is still relevant. So
Yeah.
kind of hung in there.
100%. It's probably a good place to wrap up there. Um, as always, guys, like take what we talked about today and try to apply it now in your own studying. Answer practice questions. See if you can explain it to others, and enjoy the process. But on behalf of myself, Eric, and Eric 2.0, we wish you guys happy studying.
He's waving at you. Wave.
And
Wave.
what's his name?
This is Owen.
Owen, how's it going? Oh my God. This is going to be the highest grossing episode of this podcast
These
now.
guys really wanted to be a part of this episode, so maybe another day we'll.
That's a future doctor right there.
Maybe so.
All right,
My daughter. But before we sign off, my daughter would say, "We have this book called Future Doctor." I would always say, "Belle, what do you want to be when you grow up?" And I would say, "Do you want to be a doctor?" She said, "No." And then the other day I came home and she said, "Are you wearing your doctor clothes?" I said, "Yeah." "Are you going to be a doctor someday?" And she said, "No, girls can't be doctors." And I was like, no, Belle, you can be a doctor.
Yeah.
Doctor if you want.
Girls can definitely be doctors. Some of the best doctors I've had have been amazing women. And
absolutely.
Well, oh man, that's awesome.
All right, sorry, it's turning into a little bit of a production here.
Family picture. I'm sure your wife is very grateful for your ability to multitask.
Yeah, we're doing it.
Well,
anyway,
I'll let you, Owen and Bella, enjoy your weekend. I appreciate the guest appearance. They are welcome to show their lovely faces any other time. You guys have a great weekend, and to our listeners, hopefully you guys get to watch the video as opposed to just listen to the podcast, because I promise Eric's kids are well worth it. Happy studying, everybody, and we'll see you guys on the next episode of the Jack Westin MCAT podcast.
Take care, everyone.